Patty, tell me should I start now? Okay. He's just going to put up the first slide. Okay. Good afternoon. Welcome to Zaremski Forum. These are endowed by an alum, a 1975 alum of the law school, Miles Zaremski, and his wife Elena. They are noontime talks. We will have another one shortly, that's right, from Professor Van Tassel on nanotechnology, otherwise known as gray goo. That's fine. But anyway, so today I'm talking about governing non-traditional biological experiments. Yes? Oh, you can't hear me? Cool. Oh, okay, sure. Let me know if it's too loud, though. We tend to think of science as something that happens mostly in universities and corporations. But historically, this wasn't the case. So these are a number of people who just made many notable discoveries as amateur scientists, one of whom was Charles Darwin. And a lot of biological research is conducted outside of traditional university and corporate settings. It's in people's homes, it's in their garages. Sometimes it's doing projects after hours in university and corporate labs, and that too is sort of non-traditional experimenting, moonlighting, using equipment that's there during the day, but you're doing it for your own experiments, and also in so-called community labs. And these are groups of non-traditional experimenters, a couple of famous ones in New York and in the Bay Area. They use equipment and materials that they either buy online or sometimes produce themselves. They often do that with reverse engineering. And we'll talk more about community labs in a minute. So historically, if you wanted to do genetic experiments, at least of any sort of sophistication, you had to have very expensive, very complicated materials and equipment. This is kind of traditional genetic equipment. Those of you, are any of you geneticists? Okay, so you're our scientific expert. Look familiar these things? Okay. And so as a result, because of the expense and so forth, this is pretty much what confined research mostly to the traditional realms of the universities and the corporations. But developments in gene editing technology are beginning to change this, and in particular, the new gene editing technique known as CRISPR-Cas9, which make it extremely, considerably easier and cheaper to do the kinds of things that previously required much more elaborate, sophisticated, and expensive equipment. So these techniques, CRISPR-Cas9 and others, were picked up quite quickly by researchers in non-traditional settings. And there is an international competition called iGEM, where teams can submit projects. It's kind of like an international science fair, and a number of the projects that have been submitted in the last few years in that competition involve gene editing using CRISPR-Cas9. Also, you can get gene editing equipment to some extent online. There's a fellow named Josiah Zahner in the Bay Area who sells do-it-yourself kits online, and not for fairly basic experiments, but you can do this. Increasingly, you can get a hold of the equipment online, and people are making it and selling it. So what do we call this activity outside of these traditional spaces? Some people call it do-it-yourself gene editing or do-it-yourself genetic experimentation. That's probably the most common term for it. But it's not always done by yourself. I mean, it's often done in community labs with groups of people. It's often done with colleagues or with friends. So I don't like to call it do-it-yourself because, it's not always done alone. Some call it biohacking. You may have heard that term applied to this activity, but that has kind of some negative connotations. There are significant benefits that could derive from these non-traditional experiments. New scientific advances. You have to, I don't know if you remember, but Steve Jobs and Bill Gates both did their early work in their garages. The first Apple was unveiled at a local electronics hobby club. So there's a long history of important advances being made by experimentation in non-traditional settings. So for that reason, my co-investigator and I, Ron Conlon, prefer to call it non-traditional biological experimentation, which is a mouthful, or NTB or NTBE. And we became interested in this activity several years ago. I mean, I've been working on genetics since the early 1990s. I have no science background, as many of you have heard me say, which makes it kind of odd that I would be working in genetics. But when I started teaching in 1984, law professors in health science-related areas could never get grants from the government. I mean, NIH, no NSF, they would never fund us. I mean, they do scientific medical research. We don't cure anything. We just make trouble. So, but when the Human Genome Project started in 1991, there was 5% of the budget set aside for so-called ethical, legal, and social implications research. For the first time, law professors could get funding from NIH to do, legal and conceptual legal research. And I said to myself, well, time to learn genetics. And so I got together with a colleague who was then at the medical school, Jeff Botkin, at the bioethics department, who is a pediatrician and a pediatric geneticist. We got one of the very first grants from the NIH. And so I've tried to stay sort of intelligent about what's going on. When the germline gene editing story broke, starting in 2015 with the Chinese researchers with non-viable embryos and then with Dr. Hay, I was looking around. I had written books and I had gotten 5 or 6 grants over the years, and I didn't know what else was new that I could work on. And I was reading some of the transcripts from the National Academy of Sciences conferences on germline gene editing, and people began talking about how CRISPR-Cas9 was something that you might even use in your garage. And I said, aha. And so I reached out to my colleague Ron Conlon, who is a professor of genetics at the medical school, and we explored this area and eventually got a small grant, an R03 from NIH, to investigate, think about, publish on the issues that this phenomenon raises. Well, what concerns does it raise? I mean, what should we be concerned about? As I said before, potential for major scientific advances, but is there a dark side? Especially after 9/11, these technologies, and this was, in the early days, it was before CRISPR-Cas9, but even before CRISPR-Cas9, the notion of non-traditional experimentation raised concerns that the experimenters either intentionally or inadvertently could create biological weapons. And that attracted the attention of both the FBI and Homeland Security. And a couple of times they tore apart basement labs on suspicion that they were producing bioterrorism devices. This was in 2004. There was an art professor at SUNY Buffalo named Stephen Kurtz. He and his wife Hope had been preparing commissioned works at home using, among other things, sort of biological art techniques. His wife Hope died, and her husband called 911, and her death was later determined to be from natural causes. But when the emergency people showed up at the house to take care of her, they saw all this equipment, petri dishes containing bacterial cultures and food testing equipment, and they got suspicious. Remember, 2004, 2001 was kind of fresh in their minds. So they contacted the FBI, and a big brouhaha ensued. They investigated Kurtz for bioterrorism. Eventually he and the former chair of the University of Pittsburgh's Department of Human Genetics, a guy named Robert Farrell, were indicted on mail-in wire fraud for allegedly trying to get biological organisms from a lab that wasn't allowed to sell to private individuals, and I'll come back to that. In 2008, a federal judge dismissed the mail-in wire fraud charges against Kurtz, noting there was insufficient proof, but Farrell was fined $500 after he pleaded guilty to a count of mailing an injurious article, which is a misdemeanor. In 2008, the FBI and other agencies investigated the basement lab of Victor Deeb, who was a retired chemist in Massachusetts, and he was trying to develop carcinogen-free sealants for baby food jar lids. I guess they have carcinogens in them, the sealant for a baby food jar. Anyway, he was trying to invent sealants that didn't have carcinogens. There was a fire in his air conditioner. The fire department showed up, and they went up on the second floor to shut off the air conditioner, and they found 1,500 bottles, cans, and jars, and boxes of chemicals when they went downstairs in the basement to turn off the power. The city banned him from this was in Marlborough, Mass., banned him from further experiments. He countersued against them for loss of his intellectual property and for emotional distress. I don't know the status of that suit. I haven't been able to see any recent developments. So that was kind of the initial response, was a very sort of aggressive police, law enforcement attention to it. The FBI has continued to try to stay on top of what's going on, but they have switched their modus operandi to friendly cooperation and observation, and they go to community labs and teach safe experimental practices. They do so at conferences. They even help finance iGEM, and this is Edward Yu, who is the main FBI person on this. He's the supervisory special agent in the FBI's Weapons of Mass Destruction Directorate, and this is him presenting at a meeting of these experimenters at one of their conferences. So the FBI continues to be there, but in a very sort of friendly rather than hostile stance. But there's still a lot of bioterrorism concerns, and in 2016, the United States declared CRISPR to be a weapon of mass destruction. It's the only biotechnology on the list of WMDs, and so, you know, concerns continue. Intentional or inadvertent creation of harmful agents, pathogens, weapons, and so on. Another concern is gene drives. Who knows what a gene drive is? Who can tell us? Anybody tell us what a gene drive is? Well, it's, I mean, in its simplest, sort of explanation, it's a genetic modification. So if you genetically modify, say, mosquitoes, and you release them into the environment, so you hope that, you know, if they're sterile, they will mate with other mosquitoes and there won't be a lot of harmful mosquitoes around. But the genetic modification that you make tends, for reasons of genetics, to disappear and to be incomplete fairly rapidly. So what gene drives do is they make genetic modifications in such a way that the alterations spread both faster and more permanently in populations, for example, mosquitoes. So a concern was that experimenters in their basements could either intentionally or inadvertently create gene drives, not in humans, but and the gene drive would be released into the environment and could have significant effects on organisms, plants, animals, and so on. Another concern, which Professor Nard and I thought about for a while, is the impact on the biotech industry. So some of these experimenters are sort of open source type folks. Their idea is that anything that they or anybody else like them produce should be available for free for the public. But others are kind of more commercially interested, more entrepreneurial. I mean, there are venture capital fairs where some of these experimenters come and present some of their achievements in the hopes of getting funding and hoping eventually to be able to commercialize what they do. Both groups, however, raise the risk to the traditional biotech industry of having their experiments, the results of their experiments, be considered prior art, which would prevent the biotech companies, when they make the discovery, from patenting it. So that is potentially fairly disruptive in the intellectual property sphere. I'm particularly concerned about unethical experimentation that causes harm to experimental subjects, animals and humans. You can think about, if you know about what people have thought about doing with gene editing, you can readily imagine some of the things that people might do. And indeed, if you're watching what's going on, looking at social media websites for this community, you'll see that there's a fair amount of activity, and I'll come back to that. Is there anything besides FBI oversight that's kind of keeping tabs on this? Well, the community labs have their own restrictions, their own rules of conduct. For example, they don't let their folks do experiments beyond biosafety level 1. A lot of the kinds of experiments that might produce more questionable or more results of concern are not allowed to be done in the community lab setting. In 2013, a group of these experimenters got together in the early, around 2011, I think 2010, and created an organization called the DIY Bio. That's one word. And they held a Congress in 2013, and they adopted a code of ethics. And it had some good things in it, like, adopt safe practices, and biotechnology should only be used for peaceful purposes. But that's pretty, hortatory, pretty vague, right? They didn't have any details about, what was safe and what was unsafe. They have a biosafety advisory committee group, volunteers who are available. I believe they're still active. They were a year ago when I last checked. Who are available if you want to do an experiment, if you want to run your proposed experiment past them. They'll give you their feedback on safety and efficacy. Some of the community labs have relationships with local institutional IRBs. So, for example, GenSpace in New York has some cooperation from the Cornell Weill IRB, and they will run some of their experiments informally past the IRB. But that's pretty much it, in addition to the FBI oversight. And so, you might be saying to yourself, yeah, but, there's all kinds of government regulatory agencies that regulate things like this, right? The EPA, the Department of Agriculture, the FDA. Well, the EPA regulates microbial pesticides under the Federal Insecticide, Fungicide, and Rodenticide Act. They also regulate other recombinant DNA microbes and algae that are intended for general commercial and environmental applications under the Toxic Substances Control Act. But, and this is their language, the reach of the law is limited to commercial or commercial research and development activities, and it's unclear that non-commercial research efforts by DIY users are covered. Department of Agriculture's Animal and Plant Health Inspection Service, APHIS, the little things that crawl on plants that the ants milk, anyway, regulates field trials and commercial introduction only of plants that are genetically engineered through the use of known plant pests, and they've acknowledged that they don't have the authority to regulate crops that are modified with the latest gene editing techniques. As for FDA regulates research on genetically modified food additives, and human and animal drugs and biologics. It has asserted regulatory authority over human gene therapy, but its oversight over non-traditional experiments is very murky if no commercial product results. Now, the government is in the process of revising something called the Coordinated Framework for the Regulation of Biotechnology Products to cope with new editing techniques, but it's not clear if the framework will, which is not final, will extend to non-commercial research, and in any event, it doesn't cover human experiments. NIH oversight is limited to research that is federally funded or conducted at academic institutions like ours that receive federal funding. Intentional release of an engineered bioterror agent would be a crime. People whose health or property were harmed by such a release or by any other unethical experiments might file civil suits to recover damages, tort suits. But it may not be possible to hold the experimenters accountable because we may not know who they are. They may not be subject to U.S. Legal jurisdiction. They may be outside the country and be non-extraditable. And they may not have enough assets or any assets to satisfy a court judgment. So, what to do? Well, some people say no need to worry. One of them is Hank Greeley. He's a professor at Stanford. He's been very prominent in the conversation over germline gene editing. He's a law professor. And he said in discussing the prospect of germline gene editing by non-traditional experimenters, it remains to be seen how the ease of CRISPR will impact this type of use. Garage applications are not realistic. In 2014, an entity called the Woodrow Wilson Center, which has done a lot of research and thinking about this realm, concluded that most DIYers are still learning basic biotechnology, and their sophistication is so limited that they're not, don't worry about it, they're not going to be able to do anything, and if they do, it's not going to succeed. Couple of problems. First of all, there's perhaps more to worry about from experiments that don't succeed than from experiments that do. Think about, harming human subjects. It's when we fail and we aren't careful enough. That's when often when the problem arises. Also, you have to be careful about from who is skeptical, who is saying these skeptical, dismissive things. Much of it comes from the traditional biological community. And they have a vested interest in not having additional regulation created, which would interfere with their research. And there's a very well-known article by a scientist named John Gordon a number of years ago in which he poo-pooed human gene editing in non-traditional places, and particularly germline gene editing, and said, but at the end of his article, which was, I think, in Science, he was honest enough to admit that, we've got to watch out, because if we stimulate greater regulatory response, that could chill our own efforts. And also, we have to be careful not to be too dismissive. This is one of my favorite things. Take a moment to read this. So what can non-traditional biological experimenters currently do with regard to gene editing? So here I had to defer to my colleague Ron Conlon. They can create gene drives in non-animal organisms. They can create gene drives in some vertebrates, fish and frogs. Humans, uncertain whether that could actually be done, how much gene editing can actually be done in humans. Somatic, which means that it won't be passed on to descendants, or germline, which means that it is. Some non-traditional experimenters claim that they are now able to modify human genes. Josiah Zahner claims that he's done it to himself, but their claims are very hard to verify. But in the next 5 to 10 years, my colleague, Professor Conlon, tells me that new developments may significantly advance what can be done both in traditional and non-traditional settings. These developments include the use of nanoparticle and liposomal delivery systems, which are potentially much easier to develop than the traditional way of getting an edited gene into an organism, which is using viral vectors. And also something that Conlon is particularly excited about, which is allelic drives. These are drives that can make multiple changes in genes much more easily than you can even with CRISPR-Cas9. So, do we need to be more vigilant? Do we need more than FBI oversight? And if so, what should that oversight be? And when should we implement it? When should we trigger it? Or what should trigger it? So that's kind of what Ron and I are spending most of our time thinking about as our grant progresses. This isn't the first time that we've encountered potentially risky amateur activity. So one of the things that us lawyers like to do is think of analogous examples. How have we responded to those? So, what are some examples of amateur activity that could, potentially cause harm? Well, ham radio interfering with, particularly around before and the beginning of World War I, interfering with military and governmental signaling. At the same time, it was realized that ham radio might have distinct benefits, like warning about disasters and things like that. The response has been you have to have a license to be a ham radio operator. Local cities and counties have zoning laws that restrict what kind of ham radio equipment you can have, particularly outside or on top of your home. Sport rocketry. Anybody see, what was the movie? October Sky? How many of you have seen October Sky? Oh, the time passes. Great movie about the early days, relatively early days of amateur rocketry by a young man who went on to become a major NASA rocketry engineer. What have we done about that? So obvious dangers, right? Rocket going up, hitting an airliner, coming down, starting a fire, burning, hitting somebody on the head. Amateur rocketry has been governed by, to a large extent, by amateur associations and non-governmental public safety organizations. So there's sort of these not-for-profit safety groups that have weighed in with standards and guidelines for amateur rocketry. But there has been, as you may know, federal, state, and local regulation. The Bureau of Alcohol, Tobacco, and Firearms considers sport rockets to be firearms, or at least fall within that bailiwick. And they have a limit on the amount of propellant that you can put into your rocket motor. It's 62.5 grams. More than that is illegal. And there have been challenges, and people have gotten in trouble. Another interesting analogy is home brewing. Now, it's not like, we're not particularly concerned about people, creating poisonous alcohol, although that's, but the traditional, brewing and alcohol industry doesn't want competition, and the government wants taxes, which these folks don't pay. Interestingly, the interests of the home brewers are represented by a division of the Commercial Brewers Association. So the business interests created a subgroup that supposedly fosters the interests of home brewers, and of course, has a kind of divided loyalty there. New Jersey at one point said you had to have a license to make anything alcoholic at home, but they backed off after there was a lot of pushback. What there is the ATF. Again, the ATF has a limit on how much you can make. In the news, drones, right? Obvious safety issues there. Fairly strict regulation by the Federal Aviation Administration, including that you have to register your drone if it weighs more than half a pound. Think about whether the risks that these other activities have generated, concerns that they've generated, and the way we've responded to them, are they analogous to home gene editing, and are the responses potentially useful? Finally, computer hacking. Originally, it was celebrated as a way of proofing software, right? And if they had done more of that, we wouldn't be in the position we are today, where President Trump won the Ohio caucus last night. Because of computer hacking, in 1986, the Congress passed the Federal Computer Fraud and Abuse Act, and the Secret Service began sting operations in the early 1990s. And so there's been a fairly vigorous, as you know, and unfortunately hardly successful effort to control and prevent computer hacking. This is, by the way, what the computers looked like when they were criminalized back in the day. I had one of those. If we think about non-traditional biological experiments, what interests are at stake? So one of the things you want to think about when you think about how should we govern these activities is, what are the various interests? Are they consistent with one another? Are they at loggerheads? Which are more compelling from a public interest standpoint than others? So Here's a list that Ron and I came up with. First of all, it's important that people have the freedom to experiment. I mean, we're big on freedom and liberty in this country. The advancement of science, as you saw in one of my early slides, a lot of experimentation in biology has been by amateurs, including also in other realms. A lot of scientific advances have come out of this. We don't want to prevent that from happening. Spread of scientific knowledge. So there's a thought that as these amateur experimenters become more sophisticated, they will learn how to do things, and they will teach others how to do things, and we will, as a population, become more knowledgeable about science and therefore able to make greater advances. A lot of these folks have a counterculture ethos, which is important to bear in mind. They see themselves as rebels, and that, of course, has to be thought of very seriously when we think about how should we govern this space. Financial gain, as I said, some of these people hope that they will get rich doing this. There's a concern that whatever we do doesn't unduly interfere with traditional biological interests, the interests of those folks in the traditional experimental realm who have their status and their careers, their tenure, money at stake. And so, at the same time that a lot of the dismissal of these activities comes from these traditional folks who don't want their own toes to be stepped on, that's a legitimate concern, right? These people have invested a lot of time and money getting trained, getting educated, and they have a right to not have their activities unduly restricted. Of course, the big thing, in avoidance of harm to subjects, animal and especially humans, and then finally, and of course, perhaps paramount, the public health concerns, safety and well-being. So, what are the regulatory options? What can we do in response to this phenomenon? Well, one response is the status quo. Let's continue to do what we're doing. We're doing just fine. There haven't been any known, catastrophes, really troublesome gene drive releases, pathogens, terrorism. So far as we know, nobody's died. We don't hear a lot about animal cruelty issues. So let's just continue to do things the way they are. And that's fine, except that as the technology becomes more sophisticated and more accessible to this community, the question is, will we perhaps need to do something more at some point? And you can think of whether we should be proactive, put some governance methods in place just in case or to prevent a problem, or reactive, which is, well, let's wait until there's a problem and then have something in that can be put in place fairly readily. So either proactively or reactively, what could we do? Well, greater self-governance, and I'll come back to what that might look like. Industry restrictions on access to materials and equipment. So this is really interesting. In order to use these CRISPR-Cas9, for example, you have to use some substances, particularly reagents, and it's pretty impossible to make those yourself. You have to buy them from a commercial source. And that's likely to continue to be the case for the foreseeable future, that you have to purchase these. And so the companies that make these reagents have a policy that they will only sell them to legitimate establishments, typically to the traditional commercial and university experimenters. But the non-traditional experimenters have found ways around this. Apparently, the industry, the chemical companies are not that vigilant. So long as you appear to have a company name and a company kind of address, they'll ship it to you. And so that has not been a very effective limit. If we wanted it to be more effective, how could we do that? I think the only way to do that would be to say that if you want to buy something from one of these companies, you have to show them that you have a license. And that leads to the next realm of licensure, such as we have with ham radio. And that could include, for example, training requirements. Whether or not you get a license, you could be required to go through certain training. Think of CLE. Prior approval from who is an interesting question. The FDA, some self-regulatory body, prior approval before you can go ahead and do an experiment. Licensure, it's interesting to think about requiring these folks to be licensed in order to get the the materials they need. And of course, the big problem is that, going back to the counterculture ethos that many of these people have, what you don't want to do is drive it underground, because that's where you don't get to see it. They don't show up at conferences. They don't come to do things in community labs. And that's really, so it's a delicate balancing between trying to be vigilant and having mechanisms in place to prevent bad things from happening, but not driving it underground where, like computer hacking, it becomes very, very difficult to deal with. I mentioned before civil and criminal liability for harm to subjects or the environment. I guess the sort of extreme is Germany, which in 2017 made gene editing by non-traditional experimenters a crime. Again, thinking about the need to avoid overreacting, the need to try to give these people as much say in their own governance as we can. Continuing to foster these activities because of their potential benefit to society. Ron and I have several sort of tentative recommendations, and we're in the process of sort of thinking through them more carefully. One is don't do anything yet. That is, don't do anything very vigorous or restrictive yet. Don't jump the gun. Phase additional governance mechanisms in if and as they're needed, because otherwise you're just asking for trouble driving things underground. The next step would be greater self-governance, and actually that's something that we can begin to do now. I don't think that would be overreaching. How would this community govern itself? Well, are there any entities, are there any organizations that exist that might have a role in organization? Well, you have the DIY bio type sort of pan organization, and they certainly have been, as I said, have done things and might do things again. Also, the community labs, they have a vested interest in being able to continue to do what they're doing. Therefore, it seems like they might be a good place to embody some self-regulatory expectations, right? Watch out what not only your people are doing, but what other people are doing in this realm. So self-monitoring, self-reporting. If you hear that somebody's about to, do human germline gene editing, you might want to tell somebody. Query, who should that be? How should they respond? One idea that I've heard is to create an IRB or multiple IRBs for the non-traditional experimenters to consult, just voluntarily, sort of like that science advisory group that DIY Bio has or had. And there's actually some activity on this to create a national NTBE IRB. Then there could be self-imposed, imposed by the community sanctions when they identify someone who's doing something they shouldn't do. They could be, prevented from from being part of labs, although again the risk is driving them underground. They could be precluded from competing at IGEM and other kinds of competitions. They could be precluded from presenting at conferences. And ultimately, they could be, whistleblowing to government agencies. Hey, guess what so-and-so is doing? One of the places where one can learn a lot about what's going on in this community is on social media. And so we have a genetics graduate student who is looking at social media. We did this in preparation of our grant application for the grant that we have. We had actually a law student who was a former genetics, had genetics background, Look at what was being talked about on blogs and Twitter and Facebook and so on. Interesting things. People often will in this community will describe an experiment that they have in mind and see what people think. We're doing that, and we also envision using those platforms as a way to communicate with this community. So one of the things that we intend to do shortly is to begin to present some of our thinking to this community and just invite feedback. And also to do so more formally by approaching the community labs and their leaders, and also perhaps at some of these conferences. And I actually am in contact with Ed Yu, the FBI guy, and he and I have been working on some other things as well. Finally, I think we still have to keep the FBI on the ball because, again, a lot of this, whether we provoke it or whether it just happens because of this is how people like to do things, is going to be subsurface. The FBI has all kinds of technologies that can attempt, they think, to keep tabs on this. So that I think needs to continue to be in force. So that's what I have to say. Any ideas, any thoughts, I would be very welcome to. Thank you very much. Questions, comments, brickbats? Yes, Elliot.
Do you know how countries with much lower standards for biotech, like let's say China, are dealing with it, or they even have this issue?
Well, we saw what Germany's doing, right? Europe tends to be fairly vigilant. So far as I know, China is not. But query whether the scandals, the recent scandals, will stimulate them to do more. So far as I know, we're watching the international, realms, but so far, there isn't much being done. Again, besides this rather draconian response in Germany. And also, you got to remember their regulatory systems and their legal systems are can be different, and they may have to do things differently. Yes, Professor Van Tassel.
What about I think about the cure versus the fix?
Mm-hmm.
And what about the trade association model? Is that something that you've been thinking about? In other words, for canners and packers of food, they join together, they have a national food processors association. They, because of their collective might, they're able to get lower insurance rates, that kind of thing. And then you put them together with maybe a clinic, like a law school clinic that gives you, the ability to do startup LLCs, affairs, which is something LLCs, non-profit 501(c)(3)s, get their insurance in line, get them organized so that there's a benefit and it's a free benefit. Would that potentially work in this? Be an idea?
Yeah, so I didn't mention this, and thank you for mentioning it, but we certainly have thought about incentives to do these things. So, for example, subsidizing access to equipment and materials for those who get licenses or who do comply with or cooperate with other efforts, such as other self-governing efforts. As far as the idea of LSEs and so on is, I think, most responsive to those of these folks who are interested in commercializing, capturing the intellectual property. Those folks are going to be, traditionally regulated at some point, right? They're going to produce a biologic, and the FDA is going to have oversight. Maybe not on their earliest experimentation, but certainly at the point at which things might get out of hand. They're going to be subject to pretty traditional oversight. As far as trade associations, yes. So one of the ideas about the community labs is to have them create a sort of common organization, which they kind of have very informally. DIY Bio is that kind of an organization, but ever since the two founders kind of went on to other things, it's kind of, my understanding is it's kind of fallen into somewhat lassitude. And maybe there's room for a new trade association. But again, you gotta sort of think about the people who don't want to be part of an organization, and will the trade association only be representing community lab interests, which are, somewhat different than the people who are doing this in their garages, and who eschew community labs for various reasons, in part because of the restrictions on what they can do there. Yes?
Is there any data on the background of the people who engage in biohacking? Like, do they generally have an undergraduate degree in a field or more or less?
I have not seen any formal data on that. My sense from just looking at who they are on social media and otherwise is it runs the gamut. Some of them, as I said before, are highly trained science people who in the daytime they do they work in universities and corporations. On weekends and so forth they work elsewhere or doing their own experiments. And other people are fairly newbies, unsophisticated. They just think it might be fun to make glow-in-the-dark yeast or something like that. Or, genetically engineer their kids and, something like that. Yes, Neha? Biohazard, do you have your biohazard? Yeah, I mean, actually, that's good. I have not heard a lot of people talk about, what do you do with the junk? It's more, what do you do with with the organisms that you've just engineered and released into the environment? But that's a good point. Have a ready way of disposing of those. The hazardous experimental waste bin at the local dump is a good idea, right? Needle boxes and maybe traditional research institutions in this area could have little sort of bins outside. Well, yes.
Are there known ways of, I would say, testing something in nature for these kind of people? So, for example, if you bioengineer a plant, are there existing regulations for where and how to plant it to see that nothing spreads?
Again, yes, EPA, FDA have various, have some oversight over that. As you know, there's the concern about genetically modified plants, migrating to the next cornfield and things like that. So that's a whole realm of, that's why we're really focusing more of our concerns on humans and animals rather than plants. We're not actually thinking about plants at all, even though, you can think of horrible things resulting from efforts to fiddle with plants. Okay, well, that's all I've got. If you have other thoughts, please contact me. If you're a law student, you know how to reach me. If you're not, I'm Maxwell Melman. You can just send me an email at maxwellmelman@case. Thank you for attending. Make sure you grab food if you haven't already. And I'll see some of you shortly.